Bio 30, welcome to the introduction to DNA and interphase. We may not get to interphase in this, it's mostly just about DNA, but we're starting on the topic called molecular genetics. So what we're going to talk about today is DNA and RNA, its structure, mostly just DNA structure today, who contributed to the discovery, and what is so important about DNA. By the end of this, I'm hoping you are able to tell me what makes up DNA, the backbones and the rungs of the DNA. You should be able to define the words pyrimidines and purines. You should be able to understand that nitrogenous bases match in pairs. We call them complementary to each other, so A's with T's and G's with C's. I also want you to know the three names of the key players who contributed to the discovery of the structure of DNA, as well as where it's found in the body, because we have a couple different places you can find different types of DNA, and as well as what role does DNA play within your body. So we have chemical cells, okay? Chemical codes of cells, I should say. Nucleic acids. Nucleic acids, you may remember back from Bio 20, are one of those 4 macromolecules, and we can break them down into nucleotides and then nucleosides, and we're going to talk about the differences of those in a couple days. Essentially, guys, they're really, really important to multicellular organisms, single-cell organisms, because essentially they determine the functions and characteristics of our cells. So there's two different types. There's RNA and DNA. And you can see from the picture that DNA is a double strand. So it has 2 strands, each pair matching with something: adenine, guanine, cytosine, and thymine. You'll notice adenine and guanine are a little bit bigger than cytosine and thymine. Okay, that's why A's with T's, G's with C's. Okay. Adenine, guanine, cytosine, and uracil are found in RNA, and RNA is only a single strand. We're going to learn why that's so important here during the replication and protein synthesis phases. So RNA is known as ribonucleic acids, and DNA is deoxyribonucleic acid. So DNA is the main component of genes. So inside your cells, you have a nucleus, and inside that nucleus is your DNA. And on the DNA are these things called genes. And genes are essentially what you get from mom and dad, and they're the reason your hair color is brown, your eye color is blue, you have freckles, you don't have freckles, your height, your your weight to an extent, okay? That is, the genes tell the story of you. So because they are a really important key, thanks, bud, because they're a really important key, they're never allowed to leave the nucleus. They stay there. So we need somebody, RNA, for example, to make a copy. And that's the whole point of RNA, is it's the instructions for making the proteins that make up us. These proteins are what causes the cells to function, and the reason your eye color is your eye color, the reason that your hair color is your hair color, for example. Now, this may not make sense right now, but we're going to learn about replication and protein synthesis throughout this unit, and stuff like this will start to make sense. So what is the structure and function of DNA? Well, deoxyribose, I should say ribonucleic acid, appears in like spaghetti-like structure. So think like if you had one piece of really, really, really long spaghetti, okay? And it's just kind of like mishmashy in there, and then there's a bunch of them together. These long strings are called chromatin. And tomorrow you're going to have a little bit of a vocabulary video, so you will learn a little bit more about chromatin. But essentially, chromatin is so long, we need to make it smaller and easier to read, so we condense it down into something called chromosomes. So chromatin and chromosomes are actually the same thing. They just have a little bit of a different name because they look a little bit different. If you're looking at you're writing your notes down, you'll notice that there's a picture of how chromatin condenses down to chromosomes. So you'll see that it winds tightly around these little structures, okay? We don't need to go into that. Just know that it's this long, long spaghetti-like stuff, and then it kind of comes down into a condensed chromosome. So there are a couple different types of DNA. You can have your nuclear DNA, which is what I've kind of already spoke about. You have what's called mitochondrial DNA. So the mitochondria within the cells actually have their own DNA called a plasmid. The chloroplasts found only in plants also have something very similar. So these are the 3 types of DNA. Obviously, we do not have chloroplast DNA, but we have nuclear DNA and mitochondrial DNA. Please write all of this down in your notes, so maybe pause the video at this moment, get this down, and then we'll move on. So here's some interesting facts. Mitochondrial DNA and chloroplast DNA have their own DNA because there is a theory that they were bacterial at one time. So they were a type of bacteria, and they eventually joined up with eukaryote cells, or eukaryotes. But back when we were like single-celled organisms, not us, but great ancestors, right? And they ended up living in a symbiotic relationship. And when that symbiotic relationship happened, they kept their own DNA, but then integrated into our cells. The other very interesting thing is mitochondrial DNA is actually taken from a mother and passed to all of her children. So if you were to test your mitochondrial DNA, it would be the same as your mom's, which would be the same as your grandma's, which would be the same as your great grandma's. So men don't pass on their mitochondrial DNA. So, for example, I myself pass my mitochondrial DNA onto my son and daughter. My husband's mitochondrial DNA would be the same as his mother's, but he would not pass that on. This is used to study evolutionary history and lineage. And they have tried to use this to go back to what they call the mitochondrial Eve, trying to find that very first woman. Because if it matches and you can go back and back and back and back, you can look for lineage. We're going to move on now to talking about who helped discover DNA. So there's a couple key players, Rosalind Franklin and Watson and Crick. So Rosalind Franklin, she was a crystallographer. She took X-ray pictures, and she actually took the very first picture of DNA, which then Watson and Crick used a couple years later to actually determine the 3D structure of DNA. Now, the unfortunate part was because it was in the early 50s that this occurred, Watson and Crick went on to win the Nobel Prize, while Rosalind Franklin was not given any credit for her work. It wasn't until the late 70s or 80s she was given like a posthumous Nobel Peace Prize, but unfortunately by that point she had passed. She passed quite young because she was a crystallographer and worked with X-rays. Back then they didn't understand the dangers of an X-ray to your cells and how they mutate cells, and she ended up dying quite young of cancer. So these are the three key players. You can actually just look up online a couple things about them if you want. Very interesting stuff about Watson and Crick and then Rosalind. So the structure of DNA is where I want to head to next. So the structure of DNA is, we say they're made up of complementary strands, not identical strands, complementary. Complementary because they match one another, but they're not identical, okay? Kind of think of them like fraternal twins instead of identical twins, okay? So the complementary strands are made up with the deoxyribose sugar and a phosphate molecule. And then they each have their own nitrogenous bases. Now, the reason we call them complementary is because adenine always pairs with thymine and guanine with cytosine, which means it's not adenine and adenine, guanine and guanine. No, they find their complementary pair and they pair up. If you are looking at your notes still, you will notice that at the very top, there's a picture of DNA colored, at least on mine because I have them open on my computer. And you'll notice that they match up based off of the colors. So the yellows with the pinks and the greens with the reds, if you have it open on a computer or printed it in color. But essentially, they're not identical. The one side does not match the exact with color. It's just based on the complementary. So we always say adenine pairs with thymine, cytosine pairs with guanine. So what does this mean for us? Well, this means we need to understand some key parts, which is the pyrimidines and the purines, okay? Now, had we been taking a diploma in this class, I would have said, don't worry, these are on your data sheet. You don't really need to worry about that, but you do need to know these names. So what we have here are the big ones with the smaller name. Don't you love how that works? Okay, so the big ones are, apologize, the guanine and adenine. Cytosine and thymine are smaller in structure, as you can kind of see there. And so the big one pairs with a small one. All right, so that's kind of the idea there. Now, what I want you to do is stop and practice time. There is a structure of DNA review in your notes. At this point, pause the video and try these little practices, okay? There's a label the diagram below and label a single phosphate and all that. If you're stuck, you can kind of see up above the little hexagons, okay? Those are your sugars, and the little circles are your phosphates. Anything with a pointy end is an is an A. Anything with a rounded end is a G. Hopefully that makes sense. And then you're going to kind of go through and make some matches on your DNA, as well as then you're going to have to tell me some examples from your notes. So it says, number 4 example says, if a particular strand of DNA is comprised of 24% adenine, what is the percentage of guanine? So this is just a simple matching with math. So if I have 24% adenine, I have to have the exact same amount of thymine because they're matching. So 24 and 24 is 48. Correct? Yes? Okay, good. Now all you need to know is, okay, I have 100% of my DNA. If I have 48, that means I'm left over with 52, and you're going to divide it by 2 to get your guanine. So you're going to do those questions. I will help and take questions on Wednesday of this if you are unsure how to complete that. Okay? So the final thing, I'm sorry guys, I keep getting interrupted in this video. The final thing we're going to talk about is the main functions of DNA. So the main functions of DNA is there's 2. There's protein manufacturing, and then there's the hereditary side. I'm going to tell you right now, we are going to focus mostly in this chapter on the protein manufacturing side. And then we're going to get a little bit into the hereditary side when we talk about mitosis, meiosis, but chapter 17 is pretty much all of genetics, is all about that genetic stuff. So protein manufacturing is essentially taking the DNA, copying it, and making a copy so that we can make proteins. So we're copying, what are we copying? Well, we're essentially trying to copy genes. So it's segments of DNA on the chromosome that are called genes. That's what we want. We want these sections that tell us information. You have chromosomes, genes on chromosomes that tell you, like I said, eye color, hair color, freckles, left-handed, right-handed. If you have height and whether or not some of it has to do with a little bit of hereditary, like being coordinated and stuff like that. So that's what we're looking for. So they have a variety of functions and a variety of expressions. And we are going to learn all about protein manufacturing and transcription and translation. The other thing they do is serve as hereditary material. So genes actually code for the same protein, but they can have slight differences. Okay, that's why you can have brown eyes and blue eyes. Okay, they're just a few nitrogenous bases different. So just a few A's, T's, and G's different. Okay, so that's the other important part about the hereditary material. Now, the other important part about it is it creates for uniqueness. So when we look at DNA, DNA is unique because of these couple things that occur. Now, when we copy DNA, it's not the most perfect thing every single time because there's so much to copy. So sometimes there's a slight change, what we call mutations. All right. We're going to learn this term crossing over in a little while, but essentially when you make an egg and a sperm, the chromosomes actually switch some information so that they're slightly different from each other. And then the other important understanding for the reason why you're a little bit different than your siblings, for example, is that random gene combinations, and we're going to see this in mitosis and meiosis, combine up. And then when an egg and a sperm fuse, it creates a whole different genetic code. So these 3 reasons are actually why your mom and dad don't have kids that are all identical. So if you think about your siblings, you and your siblings may look very much alike, or you may look very different, okay? And that is simply because these 3 things make sure that your DNA is not identical to your sibling's DNA, unless you're an identical twin. So hopefully you were able to follow along, get this stuff all in your notes. Again, I apologize for some of the interruptions, but if you have any questions, please let me know.